Can you hear me now? Awesome. Okay, so I was speaking about different proxies that we can use to measure quantity in the ocean. So stocks, budgets, those optics measurement we can use to say something like there's so much carbon, blah blah blah. There's so much phytoplankton carbon, and so on and so on. And this next group is the group that tells us something about quality of that biomass or mass or group of particles or group whatever constituents of the water, and it's kind of more quality. And this kind of the image here goes kind of back together. What Colin said, that when you see the variability, don't freak out. There's actually an opportunity here, and Rufus the dog is really knows that really well, because once the data start looking like shit, it might be that it's something really bad going with that. And once you take away all those doubts that the variability in your data is actually due to the fact that you have not zeroed properly, you don't have good water, whatever is left over is actually a really cool story. So don't freak out if things don't fit the pre-existing story. Maybe there's a Nature paper there, or JJR. JJR is a good journal as well. So you guys have seen this plot before. Kurt showed it yesterday, and this is a plot from Twardowski et al. 2001. If you go back in a history of optical paper publishing, 2001 was a very fruitful year. If you say BOSS 2001, there was like, what, 16 papers that he published that year? What was it? There was 5. I was looking at your website this morning. I think there was 5. And all the other ones that were like published from optics, he was a co-author. And he was co-author here as well. So there was like wasn't it? I think there was like 5 BOSS et al. In 2001. But anyways, this is the plot that you've seen yesterday, and Kurt was talking about this one. This is a really, really cool plot, because now we're talking about quality of the particles that we're encountering. Here, you see backscattering ratio. And Emmanuel was talking that one this morning. He was also talking about slope that tell us something about particle size distribution, no? And these are indices of refraction. And what I'm pointing out, this is from their study where they're looking at different groups of particles. So 1D is case 1 water, so open oceanic water. As Kurt said yesterday, this is the type of water where you have phytoplankton dominating the IOP signal. And this is the case 1 water where the particles are below 100 meter. And this is also case 1 water samples that were taking chlorophyll maxima. And what we can see, we can start reading right away cool stuff about it. We can see that based on a Young slope, these particles are very diverse size-wise. These might be a little bit smaller here. All these particles 1D, although they're spread size-wise, they all fit in the same group of index of refraction. So you can right away assume that they come from the same origin, no? But although they're the same size. Here, these guys have a lower index of refraction, which you would expect because these are chlorophyll-containing cells. They're organic, so they're very fluffy, waterful. For difference in these ones, because these are deep particles, so they're going to be like a sedimentish, smallish, not really important-ish. These are like phytoplankton detritus, and so on and so on. So using this graph, you can start to tell us a really cool story about the particle composition. So you're combining three different optical indices here, optical measurement to develop an index to say something about particle composition. And for difference, for example, this is case 2 water, bottom water. So this is sediment layer, bottom boundary sediment, some kind of bottom sediment layer, high index of refraction, high backscattering ratio, lower Young slope. And this is something that also Emmanuel spoke about. There was one of the 5 BOSS et al. Paper in 2001. We actually did this first time, suggested that there's a connection between the size spectra and the slope of attenuation. He has shown you many, many examples how has that slope being used since to infer particle size distribution. This is actually a paper of his ex-student, Slade and Emmanuel, of course, and this has been in a review, but it should be hopefully accepted soon. Is once again showing this beautiful correlation with attenuation spectral slope and size measured by LIST, which you guys are going to use today there to measure attenuation. But you can infer size from there. And backscattering spectral slope versus attenuation. So backscattering spectral slope is a very sexy measurement. Once again, why? Backscattering. Why is backscattering sexy? Exactly. You can derive from space. So we all want to see this graph. This graph is awesome. Because if it works in surface, that means we can look at the backscattering spectral slope from space and infer something about phytoplankton size. Phytoplankton size very close to phytoplankton function, and so on and so on. This is the only graph in the world measured by in situ data that is showing this relationship is done for sediment. Once you guys see that this graph is actually standing on its legs for phytoplankton composition, that is phytoplankton that is in the surface on the water, then start using it for remote sensing. Validate your stuff. But it's a cool promise. It's a really, really cool thing. It looks really great. I hope it's going to get published soon. So if you combine a couple of these particle composition parameters, you can start doing really cool stuff. So Megas Talpa, Colin mentioned it yesterday, fantastic student in optics class, Emmanuel student. He has good students. Has taken optics really, really deep. We usually do optics shell, but she's been working lately in 5,000 meters. She's trying to use optical measurements, the ones that we use in the surface water, bottom boundary layer shell things, in a hydrothermal plume. So what she's doing here, she's using the same proxies that we spoke about to infer something about particle size and particle composition of these particles in hydrothermal plume. So for example, here she's looking at the relationship of the side scatter and attenuation. So we spoke about backscatter, we spoke about forward scatter, and there's this side scattering instruments that we might talk about today afternoon. It's kind of like weird, but it's measuring different thing than backscattering, different thing than forward scatter. It's measuring side scattering, but the ratio of those two things is inferring something about particles. And it's showing that there's a great variability as we're going through the water column, up and down, in the size of the particles or composition of the particles. Here she's calculating PSD slope using the samples that she ran either in situ and LIST, so that instrument, or she calculated from using the slope of attenuation from ACS, using the discrete samples because ACS cannot be taken to this depth. And so also she did some CP profiles, and you can see the diversity. So this paper just had been accepted yesterday, so it should be coming up, and I'll put it in a Dropbox. It's pretty awesome because now you're seeing people like you coming from this class are trying to use optics in other things, but phytoplankton ecology or things like that. Another way to determine particle size is this fluctuation-based approach. And this was first time suggested by Shiffrin, who was a big physicist who worked mostly in atmospheric, and Emmanuel can correct me, in atmospheric optics. And he was looking at the fluctuations in optical signal. And as it turns out to be, the fluctuations in the optical signal, scattering, or as you can see later, chlorophyll, has to do something with the particle size. So Emmanuel, Nathan, who used to be Mary Jane's student, also took this class, and Wayne have done a set of experiments and looked at the fluctuation in backscattering and compared variance over mean ratio with the size spectra they were getting from LIST. And actually it fits really nicely. So by having this cheapo instrument, let's say backscattering instrument that you can deploy on glider out in the ocean, you can look at the variance to mean ratio with some constraint and infer something about the size of the bugs out there, size of the particles. And that's what he did. So here he took the same approach. He looked at the variance to mean ratio of the backscattering over the time of the deployment in North Atlantic. We knew that in the beginning of May, the bloom started. So the amount of chlorophyll increased. The diatoms started blooming. And we can see that from the variance to mean ratio here in chlorophyll. So as there was nothing, diatoms showed up, the variance to mean ratio of the chlorophyll fluorescent goes up. So if there's no particles, the variance to mean ratio is going to be lower. But as the particles increase, the variance to mean ratio of the chlorophyll fluorescence is going to increase. So you see there's like big guys showing up, and then they disappear. And then the small guys show up. But what we see from backscattering, we see a completely different story. Why? Because as the diatoms are dying, they're getting sticky. As they're getting sticky, they're forming aggregates. As they're forming aggregates, we start seeing them in backscattering. So by combining these two really, really cheapo sensors that you can put on a glider, not only you know something about biomass and carbon, you can say something about size and export, and formation of the aggregates and export of the carbon from the surface water. So it's pretty cool. Everything clear so far? Partially? Okay. Yes.
A few comments.
Please.
The size, beam attenuation stuff was Volt, but then Morel published articles on it before.
Okay.
Theoretically.
Okay.
Second thing, Shiffrin wrote a textbook in marine optics.
Yeah, but originally he was an atmospheric.
He talks about atmospheric.
Yeah.
He also did a book on marine optics. And this is from the same aggregation experiment. They
Exactly.
did the same exact aggregation as showed other data for was just beautiful data to test this hypothesis.
So that pretty much that experiment almost exactly the same that you.
But in the sink,
Yeah.
yeah.
So if you would do the same experiment here and measure it now during the class with the backscattermeter, you would be able to derive these relationships and use backscattering as a proxy for size. So furthermore, with that chlorophyll to backscattering ratio, so I said chlorophyll fluorescence is kind of a very, very promiscuous way of saying something about chlorophyll. We have to remember what else is carrying with it. It's carrying physiology, it's carrying species composition. Backscattering is a good thing to measure carbon if you know what you're talking about, if you have a good relationship. So in theory, chlorophyll fluorescence to backscattering ratio should tell you something about chlorophyll to carbon ratio. Chlorophyll to carbon ratio varies across different phytoplankton. And usually we know that it differs quite a lot with diatoms and flagellates. And it goes back to also to that cell size and chlorophyll and carbon. So what we observed during the same experiment that showed the slides before, the North Atlantic bloom experiment, that if we plot this backscattering to chlorophyll, that we see three different relationships as a function of time. We still first see with this relationship number 2, then the relationship number 3, and then the relationship number 4. And this kind of this number 2 was during the diatom period. This is kind of when diatoms were dying, and this is when diatoms went away. So what we took this chlorophyll to backscattering ratio shown here, and we compared it to percentage of the diatom carbon in our sample. And we found a pretty good relationship between these two numbers. So for this experiment, we know that we can use chlorophyll to backscattering ratio to infer something by by the phytoplankton composition. However, that described only the difference between groups 2 and 3 and 1. So groups 2 and 3 were diatom dominated. Group 1 was flagellate dominated. So what was driving this difference? Well, using statistics and lots of massaging and everything, we figured out this number 3, this increase in chlorophyll fluorescence, actually happened when the diatoms were dying, when they were running out of silica. And there's several theories why we were seeing that, but it was coincidental with the decrease of silica, and they're actually disappearing from the water column. So in the end, we took this ratio, and here this is a very complicated graph, but different colors. So blue is your flagellates, yellow are diatoms, and red are diatoms. You can plot community composition from different platforms, and these are gliders, and this is a float, and you can observe how does this community change or large scale parallel to the physics and different processes in the ocean. So just really fast how you can develop something from a small amount of samples, put constraint on it, and export it to these autonomous vehicles and cover really large space and time. This is something that Ali has done. This is another proxy for phytoplankton community composition, and she might mention it before, and it's really, really cool. So what she does is the composition of absorption, particulate absorption, in order to get these peaks that are associated with different pigments. She validates each of these peaks and cross-correlates them with different pigments that she has from HPLC analysis. And after she validated them, she went back and applied it to the whole Tara data set, which is really awesome. So now wherever Tara went, she can see, for example, phycoerythrin proxy distribution. So she knows where there was more phycoerythrin dominated things. So maybe there was a trichodesmium there. I don't know. There's usually trichodesmiums here. They're not there. So they wouldn't have been in Australia. But it infers something. And it really, really, once again, important thing. She validated it with a large number of data points. And I'm sure if you talk to her a little bit more, she can tell you everything about it. Going a little bit back to the CDOM stuff, some crazy stuff. So I shown before that the CDOM, the amount of CDOM in a coastal area can be related to the amount of dissolved organic carbon. No? And that, I think Colin yesterday said that you can use the slope of CDOM to infer something about composition of the dissolved organic carbon in your CDOM. Well, Helms et al., what they did, they looked at the ratio of two different slopes in UV and tried to use that to infer something about the molecular mass of the DOC in water. And this is done for New England rivers. So you can see it goes from Chesapeake up through all the way to New England here. And for all these rivers, all these samples, they found pretty strong correlation between the ratio of the molecular mass of the DOC and this, it's a ratio to slopes. So what people have started doing is start looking at that ratio of these two slopes to infer something about molecular mass of this organic carbon. So this is a paper by a group of molecular biologists who was questioning, they didn't know, they just have observed the shift in the population of iron oxidizing bacteria. So we're, once again, backing up for phytoplankton, backing up from everything. We're now in a world of iron oxidizing bacteria. Iron oxidizing bacteria, if you go in front of the lab, there's that little, behind the parking lot, there's a little pond. There's like red goo inside. That's your iron oxidizing bacteria. What happens, there's a big shift in population. They don't know why. They have no idea how that iron oxidizing bacteria lives. How does it get carbon? So they approached me to start kind of help them out. I don't know why. And I was like, well, we can measure the slope and figure out something. So by measuring the slope of the CDOM and measuring DOC, we managed to actually infer that the change in amount of DOC and type of DOC is causing the shift between these two iron oxidizing bacteria. Which is kind of bad, okay, but that actually allows them to grow now. They couldn't grow them before. Now they can grow them. They can say something about their physiology and so on and so on. So this is a cool example, once again, of how you can take optics outside of the phytoplankton and try to apply it to something else like bacterial ecology. So those are like quality things. Questions? Again, I'm just telling you stories. No? Fluxes. So fluxes, as I said, these are the sexiest things. You know, it's great to know the budgets, it's great to know the quality, but we really want to know is changes in those fluxes and changes in the quality of the particles. This was work done by Nathan Briggs, Mary Jane's student. He was also part of this class. You might have seen before. So when you do a vertical profile, let's say backscatter in the ocean, very often, so you're going to see these wiggles. And what most of the people do, they put a medium filter on it, some kind of smoothing filter, get rid of the wiggles and take whatever is left over. Because, you know, well, I don't know what the wiggles are. You know, I want nice numbers. I'm going to average 1 meter and correlate that with the rest of the world. And that's great. But what you can do, what Nathan did is, you know, take out, you know, run a medium filter, take out the signal, actually look at the wiggles. And call these wiggle spikes and assume that they're actually aggregates. Aggregates falling from that same diatom bloom that I mentioned before. So if you take all the wiggles, all the squiggles, all the spikes that have been seen by glider and group them over time and space and plot them underneath your surface backscattering or surface chlorophyll, you're going to see that these spikes actually appear during the diatom bloom. And you're going to see them kind of like falling down in space. And in backscattering, they kind of fall down and they're present up here. But in chlorophyll, these chlorophyll spikes are present just in the beginning of this export. And funniest thing is that they had sediment traps there. And when this export was seen, the sediment traps were seeing lots of fresh, still viable material. So what by looking at other people's trash pretty much, has developed a method to quantitatively say something about how much carbon, how much particles are getting exported from the surface, something about that export rate, and he can actually say something about viability of that stuff because he has this chlorophyll fluorescence, which is pretty awesome. There's another method of doing fluxes and catching this export. This is a method developed, I mean, it's a similar method was developed by Bishop et al., but Meg has modified it. She's worked with Emmanuel and some other people, and she's going to start using these floats on which usually you have 4 different sediment traps, but she has modified and removed one of these sediment traps to put attenuation meter. So what she does, she lets this float go in the ocean, lets it sit for a day on a certain depth. And as these particles fall, they fall in the optical window of your attenuation meter. So just imagine your ACS, open up the C-tube, and let things fall on it. And what you actually see is the change in your attenuation as the particles are falling on it, they're getting accumulated. So she does that for half a day moves or 1 day moves, whatever. So she can convert the increase of attenuation with the amount of particles accumulated on the window. And she can effectively calculate flux once again. So here's a plot from her paper, and you can see the change in flux measured by that optical sediment trap deployed on a float as a function of chlorophyll fluorescence in the surface and backscattering. So that's kind of pretty much it. And once again, you know, there's many entities and parameters and constituents that we can measure with optical proxies. It's just kind of flavor, a cross-section of all of them. I find great pleasure in playing with those because I don't consider myself so much optics person. I like using optics to study biogeochemistry, and this is a great way to not sit in front of the microscope. As with everything else, you really have to be careful with those. Mary Jane says that proxies work until they don't, which is really the best way of explaining them. You have to validate them. I was really trying throughout this talk show you the idea of proxy, validation of the proxy, and application of the proxy. So many times people have an idea, jump over the validation, or have a synthetic data set, which of course is easy to validate, and then you have the application. And that step missing. And it's not based on a strong and meaningful relationship with biogeochemical parameters. Also, you can have a correlation, but there's a really strong correlation between the holes in the ozone layer and the number of pirates. Is that a meaningful correlation? Not really. So, take these two things in consideration when you try and develop proxies. Interpolate rather than extrapolate. So if something works in North Atlantic, doesn't mean it's going to work in South Pacific. Rather use, collect a couple data points so you can make a good relationship and then interpolate within it, don't extrapolate. And really try to seize the variability and chaos. The irregularity in the data is usually pretty cool stuff if you can, correct for zeroing and blanks and everything that you forgot. But if you have really some variability, it can be really cool source of new parameters, new proxies that you can use and people can use. And that's pretty much it. So if you guys have any questions, I'm going to answer them. If not, I can tell you many other stories. All these papers are going to put in Dropbox, and I would recommend reading, just having them as a reference point. They are great. But again, don't use, if somebody published a carbon to backscattering relationship for Damariscotta River estuary, don't use it in tropical waters. That's one thing that you really want you to take with you. Don't do that. Measure your own carbon and develop your own new relationship. And that's it. And list handout is added, as you can see. Okay, so I think we're going to take like a 5-minute break or something, and then we're going to start the lab. And